All right, physiology students, so welcome. We're here on Tuesday night, almost the middle of October. I can't believe it. I posted a couple announcements. First of all, about this week in class. I mentioned we would go over chapter 13, parts A and B, but I kind of forgot that we still need to cover the second part of chapter 12. So I think tonight we're just going to focus on that second part of chapter 12, and we'll start chapter 13 on Thursday. And we have plenty of time to get through chapter 13. We still have a whole other week before your next lecture exam, so we have some time. So tonight we're actually doing part 2 of chapter 12, the central nervous system, so brain and spinal cord. I also extended the due date. A couple of you needed a little bit of extra time for that Physio X lab until Wednesday at midnight. So I'm not going to start grading those probably until after Wednesday, and it might take me a while to get through those. So I hope the Physio X lab went okay. This week's lab is going to be on action potential. So I kind of shove action potential down your throats because it's an important topic that I want you to focus on. So we have a Labster lab on action potential. Again, I hope it's a good review. And then if you can, remember you have 3 workshops to get through and handouts to do and then send to the MDSC by the end of the semester. And there's a link to all the workshops, and there is a workshop on action potential. If you can't get to a live session, you can watch the video and fill out the handout and send it in that way to get credit for it. But just keep that in mind, this idea of action potential, if you're still struggling with what's depolarization, repolarization, what happens in each step between the next lab and a workshop, hopefully that helps as well. And then the other announcement, I know I say I never offer extra credit, but I'm getting evaluated this semester. So I need you guys to evaluate me as a student evaluator. So look for an evaluation. It should be available somewhere in your Canvas shell after October 18th to evaluate the class and specifically me, and I will offer 2 points of extra credit, not because I want you to be kind in your evaluation, because I want you to be honest, but I'll add those 2 extra points to your upcoming lecture exam as kind of an extra credit. So I thought you guys might be interested in that. And then I'm just going to take us through our module to make sure we're all on the same page. Again, we're going through part 2 of chapter 12 tonight. It'll be a pretty kind of a review, simple part. And then next Thursday and next week, we'll do chapter 13 and chapter 14, which will be endocrine system. Here you have your lab 6, action potential Labster lab. This is the only assignment due by this Sunday. And we'll talk about what comes up next later. So I'll start. I'm just going to kind of jump right into chapter 12, part 2. And again, this is a lengthy PowerPoint. I apologize for that. I'm going to kind of skip over some of the limbic system that's at the first part of this and the reticular formation. And I'm going to jump right into these major functions of the brain, because these review slides are really important. In your upcoming lecture exam, I want you to have a good understanding of the functions of the major brain regions. And I feel like these several slides give you a good kind of summary of those functions. So here we have the cerebral hemispheres, again, right and left. They control voluntary and skilled skeletal muscle activity. They're pretty much all of the brain that you know it. And then as we go kind of more detailed into the major brain functions, we have the thalamus, the hypothalamus, and I'll kind of make this a little bigger. The thalamus relays all the sensory impulses to the cerebral cortex. So what your thalamus does, again, it's located right in the center of the brain. It receives all sensory information and it filters it and acts as like the relay station to send out that sensory information to the specific part of the brain that it should go to. It's also involved in memory processing. The hypothalamus is located directly inferior to the thalamus, and it's the chief integration center of your autonomic nervous system. So it helps to regulate body temperature, food intake, water balance, thirst. It also regulates the hormones that are released from the pituitary gland. And it also acts as an endocrine organ because it produces its own hormones from the posterior pituitary, ADH and oxytocin, which we'll get to when we talk about the endocrine system. The limbic system is involved in emotional and memory processing. We didn't spend a whole lot of time on that system. And then we have the midbrain, the pons, the medulla oblongata, these are the three kind of parts of the brain stem. The midbrain, not necessarily knowing, you don't need to know the exact specifics of each, but you should know the order that they go in. You should know that cranial nerves come off of each part of them. They contain different motor sensors. The pons contains a respiratory center that controls respiratory rate and depth. The medulla oblongata actually contains visceral nuclei that helps to control heart rate, blood vessel diameter, respiratory rate, vomiting, and coughing. So this is just a good, again, review slides of these major brain regions that you should know. The cerebellum processes all of the information from the cerebral motor cortex, visual and equilibrium pathways to help for balance, posture, and equilibrium. So that's your cerebellum. All right. And then in language, which I haven't focused on too much, there's kind of two main areas of language, the Broca's area and the Wernicke's area. The Broca's area is what helps you form words, so speech production. Patients with a lesion or some sort of disorder in the Broca's can understand the words, but they cannot speak them. So they might be able to formulate words, but they won't be able to speak them. And the Wernicke's area is involved in understanding the spoken and written words. So patients with a disorder in this area could actually speak, but the words are nonsensible. All right, so again, these are the major areas of the brain, and you should just have a general understanding of where the motor cortex is, the sensory cortex, the visual area, association area. The primary visual cortex is in the occipital lobe. So understanding the major lobes of the brain, what their major function is, and kind of just going through those major brain parts that we just went through as well will be important. I don't cover memory too much, but it is interesting. I talk about it a little bit because it's a little bit interesting to talk about the different types of memory that our brains have the ability to remember things. And memory itself is the storage of information and then the ability to retrieve that information. We have declarative memory, the memory of facts, procedural memory, the memory of skills like playing the piano or other instrument. The motor memory is the memory of the motor skills like riding a bike, which is often easy to pull out and do again. And then emotional memory is the memory of an experience linked to an emotion, like the heart pounding when you hear a rattlesnake, maybe when you're on top of a tall building. So that's kind of what memory is. I'm sure you've heard the short-term and long-term memory. These are both of declarative memory storage, so being able to retrieve facts. Short-term memory is usually limited to 7 or 8 pieces of information that you would tell someone and they maybe would forget within seconds or minutes. So that's short-term memory. And that usually decreases as we get older. My dad needs a little help with that, but you tell him something and then again and again. And then long-term memory has a limitless capacity. And it's usually easier to retrieve those long-term memories, usually because they're stored in the brain, but also because often if it's an important memory, a person has retrieved it many times to think about it. And the more often you retrieve the memory and use it, it becomes in this long-term memory and you will remember it forever. So I bring this up as well as you guys are studying for your exams. The more often you repeat this information, you read through the notes, you write your notes down, the more opportunity it has to become a long-term memory storage versus just reading it once and then trying to take an exam. So that's a little bit about memory. Again, I include these slides because some of it's interesting to go through, but I don't ask too many questions about memory. Their amnesia, which I'll let you guys read through some of this as well. We're able to detect brain waves, basically, which reflect the electrical activity of higher mental functions. And we're able to do that when we kind of hook someone up by putting electrodes on their scalp that are used to measure their brain wave activity. And this is called an electroencephalogram. And in some labs, we would be able to do this in person. You could hook your patient or your partner up to this to measure their brain waves. And there's different types of brain waves that occur in different types of activities. Alpha, beta, and theta waves, depending on the type of activity, whether you're alert, you're idle, or kind of much more common in children, for example, is a theta wave, or a delta wave is usually shown or seen in very deep sleep. So again, I don't ask you too many questions about brain waves, but I like to bring this up because it's a part of our central nervous system, the brain, and it just, again, showcases what an amazing organ our brains are. Seizures are a torrent of electrical discharges by groups of neurons. This is an epileptic seizure, and a victim may lose consciousness completely. They might fall stiffly, have uncontrollable jerking. It's usually not associated with any intellectual impairment and occurs about in 1% of the population. Genetic factors could play a role, but other brain injuries, a stroke, an infection, or a tumor could be a cause. So sometimes you hear kids with seizures, sometimes they grow out of it, sometimes there are other causes to that. Other different types of seizures, absence seizures in young children, the tonic-clonic seizure, formerly called the grand mal, this is the most severe, and this could last a few minutes where the victim is convulsing so intensely that they might break bones, lose bowel and bladder control, and severe biting of the tongue. Another thing, if someone is in a seizure, usually you want to make sure to stick something in their mouth so they don't swallow their tongue, because they usually lose all sense of the ability to control their tongue. There are anti-convolusive drugs, vagus nerve stimulator, and other therapy to try to manage those seizures. So consciousness, I'm going to kind of skip over here. We go through the sleep-wake cycle. I want to get into kind of a little bit more of what you guys will be tested on, but feel free to read through about consciousness and the ability to be conscious, the perception of sensation, voluntary initiation and control of movement. And then it goes through the different types and stages of sleep. If you're interested in sleep and the sleep-wake cycle, how sleep is regulated, we use the circadian rhythm due to melatonin released by the pineal gland in the brain to regulate your sleep-wake cycle. Usually it reflects a natural 24-hour rhythm, and then you could go through the stages of sleep over the course of 7 to 8 hours. And that's why it's important to get 7 to 8 hours of sleep, or about 7.5, to be able to get through all these stages. So again, if you're interested in learning more about sleep, I include these slides so that you can go through that if you're interested. All right, narcolepsy, insomnia, and then we'll get to meninges. So there is just so much material we could get through in physiology, and we'll focus, and I'll try to focus our studying to a few main keys. So I talk about meninges not only to talk about kind of a review of anatomy about the protection of the brain, but then we'll talk about some disorders that can be associated with the meninges. So the meninges themselves are the kind of the protective covering of the brain. So inside your skull, your brain isn't just sitting there and then covered directly by your skull bones. We have meninges that are these connective tissue layers. So these are layers that cover and protect the central nervous system. So they go around the brain as well as the spinal cord. They protect blood vessels and close the venous sinuses, and they contain cerebrospinal fluid. They also help to form some of the partitions in the skull. And they consist of these 3 layers from external to internal. You have the dura mater, the arachnoid matter, and the Pia Mater. And these are shown here. So if we look at the skin of your scalp, the periosteum is the connective tissue covering the bone, and then the bone of the skull, let's say this is your parietal bone. Then we get into the dura mater, which has 2 different layers, the arachnoid matter, and then the Pia Mater. So those are the order of the meninges in order. There are usually spaces between these layers. So if you hear of a subdural hematoma, that's usually blood forming between one of these layers, usually right underneath the dura mater. So here's the dura mater. It's the strongest meninx, that's singular for meninge, and it's made up of these 2 layers that attach to the inner surface of the skull, and then the meningeal layer, which is the true external covering of the brain. So that's the dura mater. It's the strongest. It can partition to form the dural septa, which helps to act to limit excessive movement of the brain. So it kind of is holding your brain in place and attaching it to the skull bones just to kind of keep everything where it should be. There's septa or divisions of the dura mater, which you don't need to know, but these are divisions that go usually along some of the fissures or folds of the brain tissue. Here is the dural septa, the divisions, and how the dural venous sinuses travel. So these are the veins that will help to drain the blood in the brain and how they travel through the dura mater to do that. Here's a look at a cadaver. Again, it's fun to see a real life cadaver and not just a picture drawing showing the dura mater. The next layer is the arachnoid matter. It's called arachnoid because it has some spiderweb-like extensions. It's separated from the dura mater by the subdural space. And this subarachnoid space, which is right below it, contains cerebrospinal fluid and the largest blood vessels in the brain. So that is the arachnoid matter you can see there, and you can see how it contains the largest blood vessels of the brain. Then the Pia Mater is the third, the innermost layer of the meninges. It's the delicate connective tissue layer that clings tightly to the brain, and it follows every convolution or curvature of the brain or the gyruses, and it contains many tiny blood vessels that feed the brain, and that's the Pia Mater. So meningitis, so whenever you have a word that ends in itis, that means inflammation. So keep this one in mind. You know what happens when I draw a star next to something. So meningitis is actually an inflammation of these meninges layers, and this could spread into the brain. So if your meninges layers get inflamed by a virus or bacteria, this inflammation could spread to the central nervous system, which could lead to inflammation of the brain. And we call inflammation of the brain, again, it ends in an itis word, we call that encephalitis. And meningitis is usually diagnosed by observing microbes in a sample of cerebrospinal fluid obtained by a lumbar puncture. So you might have heard of lumbar punctures before. They take kind of a needle to take out some cerebrospinal fluid in the lumbar region of the spinal cord, and they're able to measure levels of microbes in that cerebrospinal fluid for any sort of disease that could be occurring. So what is cerebrospinal fluid? This forms a liquid cushion of constant volume around the brain. It helps to give buoyancy to the central nervous system structure. So it reduces the weight of the brain, so it feels like it's floating so that it's not crushed by its own weight. So it's basically surrounds the brain so that the brain is floating in water. It also protects the central nervous system from blows and other trauma because that layer of fluid acts as another protective barrier. It nourishes the brain. It carries chemical signals. It's mostly water that's formed from blood plasma, less protein and different ion concentrations from that plasma. The choroid plexus is important because it's the cluster of capillaries within the brain that are kind of hanging out from the roof of each ventricle. And the choroid plexus is what is kind of creating the cerebrospinal fluid. So the cerebrospinal fluid is filtered from the plexus, and then you have cilia of the ependymal cells. Remember, the ependymal cells are what line those ventricles. The cilia kind of keeps the cerebrospinal fluid in motion. And you can see here that our normal cerebrospinal fluid volume is replaced about every 8 hours, which is interesting. So here's the formation of cerebrospinal fluid at number 1, showing it how it kind of travels throughout these ventricles and around the brain to form this liquid cushion of the brain. And you can read through those steps, but I won't ask you how it's formed or circulated. But these are the ependymal cells, which line the ventricles that keep the cerebrospinal fluid moving because they have the cilia on them. Hydrocephalus is any sort of obstruction that blocks cerebrospinal fluid circulation or drainage, resulting in increased pressure. And we've seen this in newborns where skull bones are unfused. Remember, newborns have soft spots or fontanelles throughout their skull so that they can fit through the birth canal. And because their skull bones are unfused, this increased pressure can actually cause the brain to enlarge past the bones. In adults, the rigidity of the skull keeps the pressure within, but this could lead to brain damage. The treatment is just to drain the cerebrospinal fluid with a shunt, that drains it to the abdominal cavity. So this is hydrocephalus, and this was common. Help me out, guys. It's the mosquito, the Zika virus, that caused this. If a woman who was pregnant, potentially got bitten by a mosquito carrying the Zika virus, she had a greater chance of giving birth to a baby with hydrocephaly, which is this enlarged skull brain. It can be, cured with a shunt to the abdomen if caught early enough. So the blood-brain barrier is another important physiological function. So again, I'm trying to focus on what I want you to learn about the central nervous system and the functions I want you to focus on. And I do want you to understand what the blood-brain barrier is. And this helps to maintain the stable environment for the brain. The substances from the blood must first pass through continuous endothelium of capillary walls, and those are made by astrocytes. So remember that astrocytes were one of our neuroglial cells that support the brain tissue, and the astrocytes have feet called perivascular feet that kind of form a tight junction around of endothelial cells around these capillaries, so that substances can move through simple diffusion or specific transport mechanism. But this blood-brain barrier does try to limit harmful substances from getting into the brain tissue that might be in the blood. So here's a look at these endothelial cells, the pericytes. Here's the astrocyte feet that kind of, again, wrap themselves around the capillary as a tight junction to kind of limit anything harmful from getting into the brain tissue. Again, nicotine and alcohol can get through this blood-brain barrier, and that's why if you smoke or drink, the effects of that you feel almost instantly because it's going right into the brain tissue. So that's how you feel it. It's also absent in some areas. So in some areas, the blood-brain barrier is absent, like the vomiting center and the hypothalamus. And that is because these two areas are necessary to monitor chemical composition and temperature of blood. And the vomiting center is a defense mechanism of your body to get rid of anything harmful. Think of food poisoning. You're throwing up because your body wants to get that out of you. Or if you have poison or anything else, these are two parts of the brain where the blood-brain barrier would not exist. So here, again, takes us through, I'm going to kind of stop this slideshow and just kind of pull out as I'm going through and picking out important information I want you to know. Traumatic brain injuries, I don't ask you a whole much questions about, but again, I included in here because I think it's important to just talk about a little bit. Concussions is a temporary alteration in function. Contusions is a permanent damage. Subdural or subarachnoid hemorrhage or hematoma is pressure from blood can force the brainstem through the foramen magnum, and this could result in death. The hematoma is when blood pools in the brain, but a hemorrhage would actually be forcing the brainstem through the foramen magnum due to lots of pressure from blood. And a cerebral edema, whenever you see the word edema, that means swelling. So this is swelling of the brain associated with a traumatic head injury. Cerebrovascular accidents, CVAs, are often called a stroke. It is caused when we have ischemia, where the tissue is deprived of blood supply, which leads to death of that particular brain tissue. And this could be caused by a blockage of a cerebral artery, by a blood clot. So this is why, if someone has blood clots, it's very scary because it could travel to the brain, form a blockage. Glutamate can act as an excitotoxin, which could worsen the condition. And hemiplasia is paralysis on one side, or sensory and speech deficits may result. So we can have transient ischemic attacks, or tissue plasminogen activator is the only approved treatment for the stroke. Again, I won't ask you guys too many questions about all the disorders, but it is interesting to note Alzheimer's disease is progressive, degenerative disease that results in dementia, memory loss, short attention span. But basically what happens in Alzheimer's is the brain cells are actually dying and then they're shrinking, and there's really no way to kind of regrow those brain cells. So actually you'll see In scans, between a normal brain and a brain of Alzheimer's, you'll actually see the brain shrinking in size as those brain cells die off, which is incredibly sad and hard to watch loved ones go through that. Parkinson's disease, the cause is a little bit unknown, but we have degeneration of dopamine releasing neurons. Because these basal nuclei are then deprived of dopamine, they become overactive. And that's what results in some of the tremors that you see. So Parkinson's disease, who's the famous actor, Michael J. Fox, the tremors, the shaking is often a common symptom of Parkinson's disease. Research into stem cell transplants is very promising for Parkinson's disease. Huntington disease is a fatal hereditary disorder caused by the accumulation of the protein huntingtin in the brain cells. What's a little scary about Huntington's disease, if you do genetic testing of yourself, oftentimes Huntington's disease doesn't present itself until later in life. So you wouldn't know if you've passed it on to your kids until much later. It's treated, but the stem cell implant research is promising. So anyway, I could talk forever about these disorders. We have different types of diagnostic procedures, CT, MRI, PET scans. These are most important for identifying tumors, areas of cancer, areas of infarct or damage to the brain. Whenever you see infarct, that means damage to tissue. So a myocardial infarct is a heart attack, damage to heart tissue. And an infarct can happen in the brain tissue as well, and that would be a stroke. Cerebral angiography uses X-rays with dye to pinpoint any stroke-causing clots, and then we'll kind of finish with the spinal cord. So we'll end with some information about the spinal cord. Again, the spinal cord, I don't ask you guys too many information about this on your test. You should remember a little bit about the anatomy of the spinal cord. It's enclosed in the vertebral column. There's different enlargements of the spinal cord, as you can see here. There's an epidural space where there's fat to kind of cushion it. Usually we do lumbar punctures or taps between L1 and L2, and we talked about doing lumbar punctures as a way to test. Let me just make sure you guys can see this. As a way to test for different microbes in the cerebrospinal fluid. So lumbar punctures or lumbar taps are important for testing cerebrospinal fluid as a way to tell us levels of certain microbes or anything else that would indicate disease. The spinal cord terminates in the conus medullaris. The filum terminale is just an extension that anchors the spinal cord to the coccyx or your tailbone. All spinal nerves are a part of your peripheral nervous system, and they attach to the spinal cord by 31 roots. There's different enlargements of the spinal cord, and then the cauda equina is the collection of nerve roots at the lowest inferior end of the vertebral canal. So here's a good review of the gross structure of the spinal cord, a dorsal or a posterior view. Here is the kind of showing the spinal nerve rootlets coming off, and the thoracic spinal cord with the Pia Mater cut open, dura mater cut open to show the spinal cord itself. And here's a look at the conus medullaris, where the spinal cord ends. This cone is the ending of the spinal cord, and then the cauda equina are all of the spinal nerves coming off of the spinal cord. All right, so I'm going to kind of skip over some of this anatomy. What I do want you to know very well is a picture that looks like this, that is labeling the parts, a cross-section of the spinal cord, and labeling the roots that go in the front and the back of the spinal cord. So here are our spinal nerves coming off of the spinal cord. And I think I'm going to go to this picture that shows it a little bit better. So we have a ventral or an anterior root that connects to the front side, the ventral side of the spinal cord, and a dorsal or posterior root connects to the back side. And in the dorsal root, we have kind of an enlargement called the dorsal root ganglion, and that's a collection of cell bodies from those neurons. The dorsal and the ventral root will join to form the spinal nerve. And what makes the dorsal and the ventral roots important is that all sensory information comes into the spinal cord through the ventral root, and all motor responses go out of the dorsal side, the posterior side. So the idea of dorsal and ventral more refers to the sensory and motor division of those roots. If we look at the spinal cord, here is the gray matter, and we have kind of a dorsal, ventral, and lateral horn of the gray matter. And then in the white matter, we have dorsal, ventral, and lateral funiculus or funiculi, which describe the columns or the tracks of axons that go up to the brain, carrying all sensory information to the brain. So here it kind of describes a little bit what I just talked about. You should know well the idea of this anatomy of a spinal cord cross-section, and the following slides kind of describe some of this for you. The dorsal, ventral, and lateral horns, not necessarily need to know what each contains. And then the ventral and dorsal roots, one's sensory, one's motor, as we go through that. All right, and this tells you the organization of the gray matter and shows how the dorsal root is all sensory information. And I totally misspoke before, I'm so sorry about that. And the ventral root is all motor information. So all sensory information comes into the back, the dorsal root of the spinal cord, and all motor information or all motor response comes out the ventral side. White matter, kind of runs in 3 directions in the spinal cord into ascending, descending, or transverse tracks. And what these are referring to is this refers to how sensory information goes up to the brain. It will travel in the white matter ascending tracks, and then it will travel from the brain to a lower cord level in a descending or a motor output. And it can also move from side to side in kind of a transverse tract. We divide the white matter into these columns, that dorsal, lateral, and ventral column. And again, just knowing that general anatomy of this is what is important for that. All right, spinal cord trauma, we can have paresthesias or paralysis caused by damage to the ventral roots or ventral horn. Again, trauma and disorders of the spinal cord, I won't ask you guys too much about it, but again, this is a little interesting. I think it's interesting, so I included in here, if you think of a paraplegia or quadriplegia, where there's a complete transection of the spinal cord, it would result in total motor and sensory loss in all the regions inferior to that cut. Polio Mellitus is this destruction of the motor neurons by the polio virus, where the muscles atrophy, especially paralysis of the respiratory muscles, your diaphragm. Again, I could talk forever about these spinal cord disorders. I think this is where it gets really interesting, but I don't ask you guys too many questions about these. ALS was the Lou Gehrig's disease. You lose the ability to speak, swallow, and breathe. That usually occurs within 5 years. All right, neuronal pathways, and I don't cover too much of this, but neuronal pathways refer to the places, whoops, and I didn't mean to do that either, the places in the spinal cord which have different tracks, and how your spinal cord brings all the information up to the brain and then back down. Decussation, so I just maybe want you to understand this wording. Decussation just means that most of the pathways cross from one side to the other in the central nervous system. And that's important to remember, because remember, your brain controls the opposite side of the body, and it also receives sensory information from the opposite side of the body. So at some point, if you sense pain or temperature when you touch a hot stove on your right hand, at some point, those sensory neurons have to cross over to the left side to get to the left side of the brain. Relay means consists of a chain of 2 or 3 neurons that are processing this signal. Somatopy is the precise spatial relationship in the CNS and how it corresponds to the rest of the body, and the symmetry just means that these pathways are paired symmetrically. So you have ascending pathways and descending pathways made up of first order, second order, third order neurons. You have common tracks that you might hear of later on. And again, ascending spinal cord tracks will always usually cross over to the other side of the body as it's taking sensory information to the other side of the brain. Then the descending paths and trackways are usually direct or indirect. There's upper and lower motor neurons involved in these, and motor or descending tracks take information from the brain to the part of the body where we want to elicit some sort of motor response or effect. So those are the descending tracts. And again, I won't ask you guys specifics about ascending or descending tracts, but I do want you to know that ascending tracts bring sensory information to the brain, descending tracts bring motor information to the body part to elicit a response, and often they'll always decussate or cross over to the opposite side of the body. And this is just showing, I'll end with this kind of PowerPoint picture. This is showing an MRI revealing just the diffusion of these fiber tracts in the brain and spinal cord. So this is showing, these tracts that are essentially neurons or nerve cells that are carrying information, so axons, to the brain and back down. How these tracts are traveling within the brain. Remember, they usually go to the thalamus first, which is located here, right in the center, because that serves as the relay point. And then that relay thalamus will send that specific tract to where it should go in the rest of the body. So I'll end kind of with this cool picture. I'll stop the recording with that.